Hello, my name is Georgios Michoulis. I'm from Greece and I am blockchain developer and data scientist. Today I will present you my master thesis at Aristotle University of Thessaloniki with the title Graph Embedding and Node Features for Drug Target Interaction, the GenGRAM-DTA approach. Let's describe two important definitions. Firstly, drug discovery in vitro is the process of identifying a chemical substance that interacts with certain biological target protein, mimicking or inhibiting its physiological activity in order to treat a disease. And secondly, drug target interaction DTI prediction is the process of predicting interactions between biological targets, proteins, and chemical compounds, drugs. Because there are more than 90 million known chemical compounds and 20,000 genes, this makes process of discovering new drugs very expensive, time-consuming, with many failures. So there is a need to reduce the search of space for wet lab validations by using in silico or computation methods. Geogenomic-based models are further categorized into five subcategories. The narrow diffusion-based methods use the drug target networks and graph-based algorithms to identify new DTIs, but also there are feature-based methods where they represent drug targets with descriptive vectors which are then used as features to train machine learning techniques. For data preparation, the first thing we did was to collect the data. The dataset we found was previously used in research work and is part of 7,800 drugs and 70,512 proteins. Also, this dataset contains drug target interaction matrix and drug-drug interaction matrix and also a protein-protein interaction matrix which were in binary form. The only negative about this dataset was that it did not include features about the drugs and proteins, and thus we had to extract them. We first collected the SMILEs for each drug from the drug bank database. SMILE is the acronym for the Simplified Molecular Input Line Entry System, which is nothing more than a representation of the molecular structures of drugs in ASCII format. Respectively, for the proteins, we collected their sequences from the UniProt database. Essentially, each amino acid is represented by a letter of the Latin alphabet, and the combination of these forms a sequence that includes information about the structure of the protein. To extract the feature from SMILES, we use the open source tools RDKit and OGB, and thus for each input compound string SMILES, we construct a corresponding molecular graph reflecting interactions between the atoms inside the compound. For proteins, we use the tool BioVec, which transforms the protein sequences into vector using 3-gram and Word2Vec methods. The left image shows the abstract overview of the GenGRAM-DTA pipeline, in which we see that target sequences are converted into vectors using the BioVec tool as described earlier. Then the vectors go through a convolution to produce the final protein embedding representation. In the case of drugs that we see in the SMILES are converted to graph using the RDK and OGB tools, which consist of molecular embeddings reflecting interactions between the atoms inside the compound as we described before. The chain convolution extracts the final drug embedding by using the previous graph vectors. The concatenation of drug and protein embedding and also a combination of linear layers produces the predicted binding affinity. RMC and MC metrics are most important for regression tags, and values closer to zero shows better performance. On the other hand, for classification tags, the most important metric is the ROC AUC, and along with the F1 score, they maximize their score to 1. We compared our metrics with GraphDTA's metrics using the same dataset. GraphDTA is a state-of-the-art project which implemented 4 different modules for DTA problem. In the comparison table, we see that GenGRAM-DTA outperformed all the GraphDTA's models. Thank you for your attention.